Hello, everyone. It's my pleasure to introduce Dr. Shan Wen as today's seminar speaker. Dr. Wen is currently a postdoctoral fellow with AstraZeneca, where he's studying population genetics to identify genetic mutations that increase a person's risk of developing blood cancer. He got his DPhil in biomedical science from University of Oxford, where he developed experimental and analytical pipelines to identify novel therapeutic biomarkers for blood cancers. Great tools like Marvel, about which he's going to talk today. Sing Sela R and Valerie has been developed by him and his group at University of Oxford, which are great tools for analyzing alternative splicing on single cell resolution. So everyone, please join me in welcoming Dr. Wen.
Okay, I will share my slides. Um, okay, let me just get this up. Um, okay, great. Great. So today, thank you for the kind introduction. So today I'll be speaking on alternative splicing analysis at single cell and tissue level resolution with Marvel. So this was a tool that I developed when I was during my PhD studies. And since then, we've continued to improve the package as well as to apply the package to various diseases. So what will I be speaking about today? So first, I'm going to convince you on why we should study alternative splicing at single cell resolution. And I'm going to go through the full functionalities of Marvel for single cell analysis. So the first would be going through the features for analyzing RNA sequencing data generated from plate-based technologies such as SmartSeq, and also to analyze RNA sequencing data generated from droplet-based technologies such as 10x Genomics. And I'm going to show you some applications of our to kind of understand the disease, the biology of certain diseases. And I'm going to show you two case studies. The first is a single cell analysis of splicing factor mutant leukemias, so blood cancers. And we'll end off with showing you an application of Marvel to study bulk RNA sequencing data. So although Marvel was developed for analyzing single cell data, it can also handle bulk or tissue level kind of analysis. So what exactly is alternative splicing? So I like to describe alternative splicing as two sides of the same coin. So what exactly does this mean? So let's say we have a gene over here that consists of various exons. And so how a gene utilizes its exons, this is what we call alternative splicing. So for example, in isoform A, you will see that it prefers to express the exon in blue, whereas for isoform B, it prefers to express the exon in pink. And for this reason, we get two different kinds of amino acid sequences, and the ultimate consequence is the expression of two different kind of proteins. And this is my favorite analogy to show over here. So if you look at the two pictures below here, it shows a picture of an apple. But if you look closely at the puzzle pieces that constitute the picture of the apple, you will see that the makeup is slightly different. And this is what alternative splicing is to sort of express slightly different versions of the same gene. Now, why should we go beyond genes and study alternative splicing? Well, I guess the short answer is that although a gene encodes several different isoforms, these different isoforms may have very different functions. And sometimes that could really mean the difference between good patient survival versus poor patient survival, or the difference between conferring drug sensitivity or conferring drug resistance. And there are also many diseases associated with abnormal splicing mechanism. So for example, over here, if you look at the schematic diagram below, splicing really only has one job, which is to remove the introns and connect the exons. And this is what we expect under normal circumstances. But there are a few ways in which splicing can go horribly wrong. So the first kind of item that could go wrong is that we get a gene mutation, or we get a mutation on a gene that is important in mediating splicing, such as the gene encoding U2AF1. There may also be an overexpression of certain splicing factors in cancer. So certain RNA binding motif proteins tend to be overexpressed in cancers. And the third way in which splicing can go horribly wrong is that we get a gene mutation um, at the splice sites. So the splice sites are typically the few base pair that comes before or after the exons. And sometimes a mutation at the splice site, such as over here, may introduce a cryptic exon. And so these are the various ways in which splicing can go very wrong and can lead to diseases such as cancer, for example. And so another reason why we should go beyond genes and study alternative splicing is because although we always begin our analysis with gene expression, such as differential gene expression analysis. So on this plot over here, you recognize the famous volcano plot that you would do when you perform differential gene expression analysis. And typically, this is a comparison between two groups. You find genes that are upregulated and you find genes that are downregulated in blue. But if you're a bioinformatician, you probably would notice that um, the wet lab scientist will ultimately come to you and ask you, so this gene is expressed. Now I want to functionally validate my gene in the laboratory. Can you tell me which specific isoform of this gene is relevant? And so, because ultimately, when we design, we design primers to validate a given gene, what we're really doing is that we're designing primers to validate a given isoform for a gene that you found upstream. And so, for example, when you perform knockout or knockdown assay, such as using single guide RNA to, splice out an exon, you have to understand what specific isoform you're dealing with, or even in overexpression as well. For example, using plasmids, when you want to build your construct, your gene construct, what you're ultimate building is not so much the gene, but the isoform sequence that we are building the construct for. And finally, for qPCR validation, just trying to understand which genes are up or down regulated. Ultimately, as I've mentioned, we're designing primers not so much for genes, but really for specific isoforms. And so for this reason, going one step beyond genes and to understand which isoforms are dysregulated in your dataset is important. Now, so hopefully I've convinced you on why we should go beyond genes and to study splicing. This slide is really trying to convince us why should we study alternative splicing at the single cell resolution. So in other words, why can't we just look at the whole tissue, but instead why should we go one step further to look at splicing at the single cell level? So if you look at the top panel over here, performing a whole tissue or bulk analysis is almost like kind of looking at the whole tissue in aggregate. And we know tissues are very heterogeneous in the sense that tissues are made up of different cell types. And so as a consequence, the signal that you get is really an aggregate or really a mixture or combinations of these different cell types. And it's really difficult to disentangle, which signal is coming from which cell types. Whereas for single cell analysis, you will see that the resolution is much better in terms of delineating the different cell types. So cell types in green, blue, red, orange, yellow, and pink. And so the signals, you can sort of attribute each signals to their respective cell types. And so why is it important to understand which signal comes from which cell type? If you look at the hierarchy of cancer stem cells on the right-hand side, you would see that cancer stem cells tend to be that group of cells that gives rise to the downstream dysfunctional or progenitor cells. And it is the cancer stem cells that continue to rejuvenate and renew themselves and lead to disease progression and even relapse. And so by identifying the signals that originate from the cancer stem cells, we can start to pinpoint which gene or which isoforms are relevant for targeted therapy. Great. So that brings us to the main part of the talk today, which is to introduce you to this R package called Marvel. So Marvel is available for download from GitHub. And so I would encourage you to use the version on GitHub rather than the version on CRAN because the version on GitHub has the latest features and bug fixes, as well as there are tutorials as well for you to go through how to prepare the input files and how to analyze the data and make sense of the data. Great. So the first main part of this talk is really diving into showing you the full functionalities of Marvel that you could use on plate-based technologies. And so the reason for showing you the full functionalities is because while there are many functionalities of Marvel, not all of them may be directly relevant to you. Maybe you're just interested in just a subset of the functions. And so for this reason, I'm showing you the full functionalities so that you can go back and sort of to evaluate which functions are the most relevant for your study. So what exactly is plate-based technology? So single-cell RNA sequencing data can be generated from plate-based methods. And so plate-based methods are typically reactions that happen at the plate level. So each well of the plate typically corresponds to one cell. So plate-based methods are typically quite labor intensive, but they typically give you good coverage, and it's probably the recommended platform for single-cell alternative splicing analysis. And so there are various methods that you could use to generate this sort of data. So SmartSeq would be probably the most popular method that you might have heard of and probably is the recommended method to go with. Great. So in this slide, I'm going to show you the A to Z in terms of the kind of features that you encounter in Marvel. So like every R package, it always begins with reading in the right type of input files. And so this step is absolutely crucial. So once we get the formatting for the input files correct, Marvel will simply read the files in and it can, in almost a semi-automatic fashion, perform the downstream analysis for us. So the input files would typically be your sample metadata, your gene metadata, your splicing coordinates, and as well as your gene expression matrix. The reason why we provide gene information as well as splicing information is because it's good to sort of analyze these two omics together, as we sometimes we like to make a comparison between these two omics. And so the next step in Marvel is that it does a QC in a sense that it tries to identify true splicing events from the false splicing events, because the last thing you want is false positive to come out of the analysis. And so there are seven main flavors or seven main types of alternative splicing events that Marvel analyzes. So the first is a skip exon. The second is mutually exclusive exons. The third is retained introns. The fourth and the fifth are alternative 5' and 3' splice site. And the last two are alternative first and last exons. So as you can see, the kind of isoforms that we detect and quantify and analyze are exon level splicing events rather than full-length isoform expression analysis. And there's a very simple reason behind this is because the RNA sequencing data that is generated by these platforms are short read. And next, after we've detected a confidence set of splicing events, the next natural thing to do is to quantify these splicing events. So just like how we quantify gene expression, we also have to quantify isoform expression. And so the unit of measurement for splicing here is percent splice in. So what this simply means is that if we have a percent splice in value of 0, it just means that all molecules in that cell doesn't express that particular exon. If we have a PSI value of 50%, it simply means that half of the molecules in that cell expresses the pink exon. And of course, you might have guessed if we have a PSI value of 100%, it simply means that all molecules in that cell expresses the exon of interest. And so once we have quantified our splicing events, we then can proceed with the downstream analysis. And of course, the most exciting part is always the clustering analysis because it kind of gives an idea on whether your cell types that you included in your study are quite different or quite similar with each other. And so we can do this in three flavors. You can do this in a standard PCA. We can also do this in t-SNE or in UMAP. The second downstream analysis is modality classification. So modality is just a fancy name for splicing pattern. So you can see that the splicing pattern can come in 7 different patterns depending on the percent splicing distribution in your cell population of interest. So for example, the included splicing pattern simply means that most cells expresses the pink exon of interest. The excluded splicing pattern simply means that most cells do not express that pink exon of interest. The bimodal has always been a very interesting pattern. So the bimodal simply means that 50% of your cell expresses the pink exon, whereas the other 50% doesn't express the pink exon. The middle modality classification simply means that every cell in your population of interest expresses half the molecule will express the pink exon and half the molecule will not express the pink exon. And finally, multimodal is that meshilineous or that other category where we see a random pattern of exon expression throughout your cell population. And so here I'm showing you that the resolution that you can get here is at the single cell level. But if you to look at the bulk level, you will see that the bulk level, as I've mentioned, is an average across all cells in a population. And if you look at the bulk or whole tissue level, it will simply be impossible to distinguish the bimodal, middle, and multimodal pattern. So this highlights one advantage of looking at splicing pattern at the single cell level rather than at the whole tissue or bulk level. And of course, the most exciting part for any RNA sequencing analysis is always the differential expression analysis. And so Malva is able to do differential splicing analysis. It's also able to do differential gene expression analysis. And so we can combine the results from both omics to understand the gene splicing analysis. So for example, a coordinate gene splicing analysis simply means that the splicing event and the gene are downregulated in the cell type in blue relative to the cell type in red. Opposing gene splicing relationship, as you might have guessed, is when, for example, the gene is upregulated in the blue cell type, but the splicing event is downregulated in the blue cell type relative to the red cell type. Isoswitch simply means that there's no differences in gene expression, as you can see at the box plot over here, but there is differential expression of the splicing event. And this isoswitch classification is an interesting classification because it tells us that there are some isoforms where you can't detect at the gene level because the gene is not differentially expressed, but you can only detect it at the isoform level because the isoforms are differentially expressed. And finally, while differential expression analysis sort of assess individual isoforms, functional annotation sort of group the list of differentially spliced genes and to give us a list of pathways that these sort of spliced genes are involved in. And another functional annotation feature that we have is nonsense-mediated decay. So what exactly is nonsense-mediated decay? On the top panel over here, you will see a normal gene being transcribed and translated into its full-length amino acid sequence. But sometimes a runaway exon may be expressed together with the gene. And sometimes this runaway exon introduces a premature stop codon, and a premature stop codon would ultimately lead to the decay of that particular transcript. So this is what we mean by nonsense-mediated decay. Great. So while this slide is helpful as a one-slide summary, it's still, I will admit, rather conceptual and rather theoretical. So what I'm going to do in the next 2 to 3 slides is to really show you some real-world scenarios in which these functions are applied. So I'm showing you here a comparison of induced pluripotent stem cell against endoderm cells. And over here, we first perform the clustering analysis, and you can see that if we look at the clustering on the left-hand side, which are based on non-differentially expressed gene, we would expect the cell clusters to not separate from each other on the PCA space. But if we take the splicing events from this list of non-differentially expressed genes, you will see that they separate quite nicely. This says that splicing is an additional source of complexity underlying gene expression. We also can perform differential splicing analysis and rank the list of splicing events that are most differentially expressed between the two cell groups on the top here, all the way to the bottom. And a comparison between those two cell types tells us that there are about 1,500 splicing events that are differentially expressed between these two cell groups. And also we can sort of input the list of 1,500 differentially splicing events, take their list of genes and do a pathway enrichment analysis. And you can see that these are the list of genes that are differentially spliced. And finally, looking at some examples of gene splicing analysis, over here, we will see that DHX9 gene is downregulated in endoderm cells relative to the stem cells. And for a selected splicing event, this is also true, where it's downregulated in the endoderm cells relative to the stem cells. As for the opposing gene splicing relationship, I've chosen to show here the BCL-AF1 gene, where the gene is downregulated in endoderm cells, but the selected splicing event is upregulated in the endoderm cells. And finally, for isoform switching, we see that for the CLAF1 gene, its gene expression is not differentially expressed between the two cell types, but if we look at its splicing pattern, you will see that it's quite different between the two cell types. So all this just suggests that there are certain genes that you can detect from using gene expression analysis, but there are certain isoforms that are differentially spliced that cannot be detected at the gene level. And so all this is nice, to have a list of splicing events that are, differentially expressed. But ultimately, we want to ask ourselves, what does all this mean? What is the consequence, or what is one of the consequence of differentially spliced gene? So one of the consequence is nonsense-mediated decay. So just to reiterate what this means, if we have under normal circumstances, a gene would be transcribed and translated to give rise to its full-length amino acid sequence. Whereas if there was, let's say, an intron retention over here, this sequence might introduce a premature stop codon right in the middle of the transcript. This leads to a translation of a truncated amino acid sequence, and this truncated amino acid sequence might give rise to a weird protein that will lead to its decay. And so over here, I'm showing us that you will see that the gene expression for genes that has been subjected to nonsense-mediated decay typically is lower. And so this reflects biology in a sense, where when a gene is subjected to nonsense-mediated decay, its expression is downregulated. And so over here, I'm showing you a function that Marvel can test, is that it plots the gene expression profile, and it annotates which genes are subjected to nonsense-mediated decay. And so you can see over here, the top left quadrant shows a list of genes that is downregulated in endoderm and also subjected to nonsense-mediated decay. And this could be a handful of genes that you might want to prioritize for your functional validation. Great. So that was for a plate-based application. The second application is for RNA sequencing data generated from droplet-based technology. And so what exactly is droplet-based technology? I'm sure you already might have heard of 10x Genomics. So 10x Genomics is the leading service provider for RNA sequencing data generated from droplet-based technology. So what this technology does is that it simply partitions each cell into a single droplet, and each single droplet has its own, chemical reaction that amplifies the cDNA, and these cDNAs will then be subjected to next generation sequencing. And so over here, the application for Marvel on droplet-based single-cell RNA sequencing would be on stem cells. being differentiated into cardiomyocytes across different durations, so 2 days, 4 days, and 10 days old. And so we begin by differential splicing analysis, and you can see that there are several genes, specifically 575 splice junctions that are upregulated in cardiomyocytes day 10 versus stem cells. And there are 243 splice junctions that are downregulated in cardiomyocytes day 10 versus stem cells. We can also do a pathway enrichment analysis. So take the list of genes whose splice junctions are differentially expressed between the two cell types and just ask ourselves which biological pathways are enriched in this list of genes. And you can see over here, the top genes that are differentially spliced are related to muscle functions. And that's not surprising because these, after all, are cardiomyocytes. So these are cardiac muscle cells. And of course, we will also like to look into the gene splicing and the relationship. So over here, we would see, for example, if you look at the TPM2 gene, you will see that its expression is quite similar between the two cell types. So the expression is quite similar between the cardiomyocytes at day 10 and the stem cells. Whereas, for example, if you look at the splice junction number 1 of the TPM2 gene, is upregulated in the cardiomyocytes day 10, but downregulated in the stem cells. For splice junction number 2 for the TPM gene, you will see that's upregulated in the stem cells, but downregulated in the cardiomyocyte cells. So then again, the message here really is to show that there are certain isoform expressions that are differentially expressed between groups of cells that can only be detected at the isoform level, but not at the gene level. And so one thing nice about Marvel is that it's able to sort of to zoom in into selected genes that you're interested in. And this could be genes that you already have an idea even before you start the experiment, or this could be a gene that could have popped up from the volcano plot, for example. So here I'm showing us the TPM2 gene again. So here we can see the gene is progressively upregulated from the stem cells to day 2, to day 4, to day 10 of the cardiomyocytes. But how does it look like for its individual isoform? So if you look at isoform number 1, it seems to be most upregulated in the stem cells, and then it gets progressively downregulated from day 2 to day 10. For splice junction number 2 or isoform number 2, you will see that its expression is quite constant from stem cells to day 2 to day 4 to day 10. For isoform number 3 or splice junction number 3, you will see that its expression is the lowest in the stem cells, but it progressively increases from day 2 to day 4 to day 10. So this again, sort of to bring home the message is that different isoforms from the same genes can have very different expression pattern relative to its gene. And so sometimes it's just nice to see where all these isoforms sit, isn't it? So you can see that splice junction number 1 sits at the both 3' end of the gene. Splice junction number 2 sits next to splice junction number 1. And the same for splice junction number 3, it sits next to splice junction number 2. And you can see that the expression of the splice junction is the highest as you go from the 3' end to the 5' end. And this is also reflected in the dot plot above as well. And the very simple reason is because this is a 3' bias sequencing where the DNA is captured from its 3' end. And so for that reason, you tend to have a bias of expression where you get the highest expression for splice junction located at the 3' end relative to the 5' end. So this is a last case study on the application of Marvel on droplet-based single-cell RNA sequencing. So over here, we've analyzed more than 100,000 cells from brain tissues of patients with autism spectrum disorder. And you can see over here nicely laid out are the different cells that you will typically find in an adult brain. And so we first performed differential splicing analysis, and we found 691 splice junctions that are upregulated in individuals with autism, and 1,903 splice junctions that are downregulated in individuals with autism. And so over here, I want to highlight this gene that consistently pops up in my volcano plot, which is SYT1. And we can see that if we look at SYT1, its gene expression is very much similar between the controls and versus individuals with autism. But if we look at the splicing profile, you would see that it's down-regulated in autism relative to individuals without autism. And so then again, the same message. There are certain isoform expressions that can only be, delineated at the isoform level, but not so much at the gene level. And pathway enrichment analysis of the list of genes that are different, list of genes that are differentially spliced between the controls and autism shows biological pathways associated with neuron development or brain development. And so this is a nice positive control to say that we are detecting genes that are relevant to autism disorders. Great. So that was the full functionalities for Marvel. If you were to apply it on RNA sequencing data generated from plate-based and droplet-based, here I'm going to just very briefly show you some selected application of Marvel, just to give you some real-world scenario of the kind of insights that you would get by studying splicing using Marvel. So the first case study I'm going to show you is single-cell analysis of blood cancer patients with splicing factor mutations. And so the specific disease that I'm going to just show you the application is myeloproliferative neoplasm. So myeloproliferative neoplasm, or MPN for short, is a kind of blood cancers. And this particular blood cancers has three main subtypes. So the first subtype is where the patient produces too much red blood cells. The second subtype is when the patients produce too much platelets. And the third subtype is when you start to observe, some fibrotic tissues in the bone marrow. And so why is this disease so relevant for us to study splicing in, or at least to show you the application of Marvel? And the reason is because in half of the individuals with this disease, we find mutations in splicing factors such as SF3B1, SRSF2, and U2AF1. And so just to remind ourselves, the splicing mechanism has one very simple job, which is to remove the introns and to give us the right combination of exons to give us a functional protein. And so over here for today's case study, I'm going to show you where the application of Marvel, where we sort of review certain dysregulated splicing in individuals with U2AF1 mutation. And U2AF1 mutation typically comes in two flavors. The first is a mutation affecting the 34th amino acid residue, and the second mutation affects the amino acid residue 157. And so over here, the list of individuals that we've included in our studies. And for each individuals, you can see that we have anything between 5 cells to, well, more than 100 cells. And these cells consist of the hematopoietic stem cells, which are the cancer stem cells. And so you will see that the different individuals would have different proportion of cells with different mutations. And because we are primarily interested in U2AF1, our patients would have, of course, the U2AF1 mutations, but they will also have other mutations that we typically find in blood cancer patients, such as JAK2 or CALR. And so for differential splicing analysis, we found 247 exons that are upregulated in the S34 patients and 176 exons that are downregulated in the S34 patients. For the Q157 patients, we found 483 exons that are upregulated in them and 531 exons that are downregulated in them. And so you would think that, oh, okay, the two mutations affect the same gene, which is due to AF1. Could this also mean that they lead to the same kind of, splicing events that are dysregulated? To our surprise, if we plot a Venn diagram, looking at the overlap between the exons that are dysregulated in the S34 versus Q157, you would see very, very, very little overlap. And this suggests that although the mutations affect the same gene, they certainly lead to very different list of exons that are dysregulated. And so when we did a PCA analysis, this is exactly what we saw. We saw that the S34 in red clusters separately from the cells of Q157 in green, which in turn cluster quite separately from the cells which don't have any mutation in U2AF1. And so here I'm showing you one gene that we thought it was interesting to sort of to look in slightly more detail, which is called GNAS. So GNAS is a gene that is recurrently mutated in blood cancers in about 1% of patients. And so it was quite interesting, therefore, to look into this gene because it's an oncogene that is not only mutated in blood cancer patients, but now we found to be differentially spliced as well. So how is this gene differentially spliced? You will see that compared to the wild type cells, the S34 cells tend to upregulate this particular exon for this gene. And so over here, I'm showing you a visual validation of this gene. So because sometimes it's just nice to sort of to visually validate your gene before going on to the laboratory to validate the genes, just in case it turned out to be a false positive. So over here, if we look at the sequencing coverage for this particular exon in orange for this gene, you will see that there are more sequencing coverage for the cells corresponding to the S34 cells relative to the wild type cells. And this is a really nice mirror for this violin plot over here, where we see it upregulated of this exon in the S34 cells. Of course, the top plot is very busy, and sometimes it's just nice to collapse this signal. And so when we collapse this signal, you will see that the mean exon expression is higher for the S34 cells relative to the wild type cells. And sometimes it's just nice to have a p-value attached to this graph over here. And you can see that the exon expression is significantly different where it corresponds to the alternative exon in orange, but not so much for the flanking exons as we would expect. And the last application that I'm going to show you is it's not single cell, but at the tissue level or bulk level. So over here, we've applied MARVEL to sort of to understand the biology of type 1 myotonic dystrophy. So what exactly is type 1 myotrophic dystrophy? So it's from the Greek word, well, I would attempt to pronounce it as dystrophia myotonica. So therefore, the acronym BM came from the Greek word. It is characterized by progressive muscle weakness and wasting, and it's typically caused by extended repeat sequence at the end of the DMPK gene. So what does it mean? So if you look at the sequencing coverage at the DMPK gene, you will see that in the unaffected individuals, you don't see a lot of coverage at the expanded repeat regions. Whereas for the DM1 gene, you would see a higher sequencing coverage at the expanded region over here. So this just shows us visually that the DM1 patient has expanded repeat regions, and this typically leads to the phenotype of muscle wasting and weakness that we see in these patients. And so how is this related to splicing? Well, so over here on the top panel, you will see a DMPK gene that has short extended repeats, but the bottom panel shows us a scenario of patients that has long repeated regions. And when there is a long extended repeat regions over here, it tends to attract and absorb this protein called MBNL1. And so MBNL1 is involved in splicing. So what we did over here was that we designed a CRISPR-Cas13 so that it binds to the extended repeat region of the DMPK. And so when these molecules bind to DMPK extended region, MBNL1 can no longer bind to this extended repeat region, and it can go on to perform its normal splicing functions. And so my colleague looked into treating these cells with the CRISPR-Cas13. And over here, I'm showing you the splicing index, which is simply the PSI value or the percent splicing value for this exon 21 or SOS1. And you can see that in a normal individual, like potentially you and I, you would have a splicing index of around 80. And in individuals with this particular disease, you will see that they have a smaller splicing index of around 30. And so over here, when we treat the cells with CRISPR-Cas13, we can rescue this particular gene in the sense that we can bring up its exon expression to that of a normal individual. A second gene is INSR, exon 11. And you can see that in unaffected individuals like you and I, you typically get a splicing index of around 15. And for individuals who are affected with this disease, its splicing index is almost close to 0. But with this treatment, we were able to bring up the splicing index to the levels that we were seeing in an unaffected individual. And so all these splicing values were computed and quantified by Marvel. And so just a quick recap. Hopefully I've convinced you why we should study alternative splicing and why we should study splicing at the single cell resolution. I show you the full functionalities of Marvel. So in other words, what Marvel has to offer in terms of all of its functions for RNA sequencing data generated from plate-based and droplet-based. And I show you further applications of Marvel just very briefly, just to give us a taste of what you would get in the real world when we perform the analysis at the single cell level and when we perform the analysis at the tissue level. And so thank you for your attention, and I'm happy to take any questions.
Maybe I can get started. So thanks, Sean, for the great presentation. I really enjoyed that. So I was just wondering if you could elaborate a little bit more on how you distinguish between the true positives and the false positives in your Marvel pipeline, the different isoforms.
Yes. So coverage is king, isn't it, when it comes to sequencing? So evidence would come from coverage, obviously. And so over here, if we were to zoom in, you will see that for, let's say we're interested in this pink exon, because this exon is alternatively spliced. And so evidence for the expression of this pink exon has to come from 3 different splice junctions. So this one over here, splice junction number 1, splice junction number 2, and splice junction number 3. Only when the exon is supported by the relevant splice junctions do we say that it's a truly expressing isoform. But let's say we encounter, an exon that's only supported by one side over here, then we know that, oh, probably it's not true.
Gotcha. Okay, thank you.
I have a follow-up question. So does the tool just need one evidence for those reads, or does it need a threshold of reads to say that it's a true splice junction?
So of course, theoretically, the minimum number of reads we would like is 1. So 1 each over here. But then again, it all boils down to your sequencing coverage. If you have really good coverage for your cells, and that's defined by 2 million pair end reads or more, then you can set that threshold to 10 reads. So in other words, it needs to be supported by 10 reads here, 10 reads here, and 10 reads here. But I have encountered data sets where coverage is not perfect, and so I lower that to 5 reads, and I think 5 reads is where you want to keep that at the lowest. If not, you're just going to be detecting a lot of junk.
Okay, so that we can adjust that threshold in the tool somewhere.
Absolutely, there's an option to do that. Yes.
Perfect. Thanks. Are there any more questions? If no more, then thank you so much, Dr. Wen. It was a great talk.
Okay, you're welcome. My pleasure.
Thank you.
All right. Thank you. Bye.